All right, so while Greg gets set up, I will just introduce our next speaker is Dr. Gregory Johnson from the Allen Institute for Cell Science over in Seattle.
I'd like to, before I get started, I'd like to thank the organizers for allowing us to come here and present. So I'm Greg. I'm from the Allen Institute for Cell Science. I'm here with a former CPC, or I'm a former CPCB alum, or I am currently an alum. I'm here with Rory, who is also an alum, and he and I work together at the Allen Institute for Cell Science, thinking about how we can model all of the data that we're generating over in Seattle, and how do we build coherent models of how cells are organized and how all of their parts fit together. And I'm going to tell you guys a little bit about an idea that we've been kicking around, which is this universal translator for biological data. And it sounds kind of crazy, and it is kind of crazy, and we've done some experiments and they work. So we're going to keep running this thing into the ground as hard as we can. And I'm hoping to convince you guys that this is a good idea. And if you don't think it's a good idea, I really want to hear why. So for those of you who don't know what a babblefish is, it's a small yellow leech-like creature, probably the oddest thing in the world. It feeds on brainwave energy, absorbing all unconscious frequencies and then excreting telepathically a matrix formed from the conscious frequencies and nerve signals picked up from the speech centers of the brain. The practical upshot of which is if you stick one in your ear, you can instantly understand anything said to you in any form of language. The speech you hear decodes the brainwave matrix. And this is a plot device from the science fiction novel Hitchhiker's Guide to the Galaxy. And there's sort of this idea of universal translators, and we've seen things like what Google has been putting out. And we want to build one of these things for all of the data types in the biological universe. And I'm going to tell you a little bit more about what that is. But this is sort of a landscape of some of the types of biological data. And this is specifically sort of like high throughput, single cell biological data, such as imaging, RNA-seq, ATAC-seq, high-C, 3C data, and FISH. And you guys have probably heard about most, if not all of these. And all of these data types are connected or can be connected in a variety of ways. Does this laser pointer work? Yeah, so for example, imaging data and FISH data can be acquired in the same cells. So I can get very sparse counts of gene expression in cells, but I can also image some subcellular structures inside of those cells and say, do some sort of analysis and say, how does the organization of the cell vary with these genes and vice versa. I can take my FISH data and relate it to less sparse, but maybe different types of artifacts, noise artifacts, RNA-seq data, and say, how, if I knew, some subset of genes, could I figure out how the rest of the gene expression varies? And then find sort of an orthogonal set of genes to run for my FISH experiment, and then relate that to cell morphology or organization. And I can do this in all sorts of ways and in all sorts of directions, and I can vary the cell lines and experimental conditions of this. And what we want, sort of as an end state for biology, is to basically say, this is what I have, tell me what I know, and tell me what I don't know. To zoom in on sort of how some of this data may be structured, such as this imaging, FISH, and RNA-seq data, we can construct a matrix and on one axis of the matrix are individual cells. So imagine that there's many, many rows here, and these are measurements from single cells. Then I can acquire different types of measurements. So in live cell images, I can take a bright field or a transmitted light image that's free. We can image the cell shape, the nuclear shape, different subcellular structures. And some of these are sort of like mutually exclusive because of the limitations of fluorescence microscopy, for example. But I can relate that morphology and some of that morphology and some genes together with fish. And I can relate those genes together with RNA-seq. And you guys are smart, and I imagine that you guys can think of many, many ways to fill in the holes in this matrix with this incomplete data. And often with these types of structured data, this takes maybe like a PhD student lifetime to figure out a way to do this in an intellectually satisfying way. And we're proposing not doing it in an intellectually satisfying way, but instead doing it in a sort of a black box way such that we can fill all of these things out and not really care how it works, but get something that gives us a good enough answer that we can use this model for a variety of interesting tasks. And we can further, as I mentioned earlier, imagine that this matrix extends in other directions. It's now a tensor. There's a dimension that's like, what type of drugs are these cells on? What type of cell types are there? Time points, so on and so forth. Just to show that this data acquisition problem is maybe not as big of a problem as we thought it was, there are many attempts or successful attempts at gathering massive amounts of high throughput data on single cell and population levels. So for example, next generation connectivity map has 1 million profiles where they vary gene expression, cell line, and perturbation. The cell image library is a collection of fluorescence images of U2OS cells that have been exposed to 30,000 different types of compounds, and so on and so forth. And all of these data sets tell us something about natural or unnatural or perturbed variation in cell biology. And we can leverage all of these types of data, stitch them together into sort of a tapestry, a continuous tapestry of single cell variation. Why do we think we can do this now rather than years ago, or actually 5 years from now? We're hoping to start now and get it done in 5 years. So there's, as I said, there's many types of these rich connections available. There are new tools, some of which I'm going to talk about, for relatively simple ways of modeling relationships between different types of data. So ordered and unordered sequences, images, movies, structured matrices. We can build models that relate these things, all of these things right now. And it's relatively straightforward. You need to be a PhD, but many of us are, and many of us are going to be. Statistical learning. Population cell linkages across conditions and time already exist. Statistical learning is really easy. Everyone does machine learning. It's a piece of cake. And so all of these things, are evidence to the fact that we can be building this type of universal translator. To give you guys an example of how this would work, so I have my Babelfish, and I have an image of a cell, and I say, okay, Babelfish, give me gene expression. And you show it this image of your cell, and the Babelfish says, okay, well, here are all of the genes that we care about, and This is the expression level of all of those genes, and I have some level of uncertainty associated with that gene expression based off of the evidence that I've given the babblefish. And then I could say, oh, okay, well, I'm going to run in the same sample a fish experiment and get some counts of some genes. But this is very, very sparse representation of the transcriptomics landscape of this sample. But that's OK, because I can update the model. So I say, OK, here's some more evidence. The model updates the gene expression levels with my observations. And the variation of some of these genes massively shrinks, because we know that those genes, some of these genes are associated with the genes that I measured. And so we're more confident in what the outcome of this question could be. Oh, and then I was like, oh, yeah, I forgot. Let me tell you a little bit about the experiment that I ran. And the model says, oh, OK, well, I've seen this type of thing before. And my confidence interval shrinks even further. And so with this fantasy universal translator that we're trying to build, we imagine that you can ask it many, many types of questions. And this is an example of a certain type of input and a certain type of output. But we want to build this to take any type of input and output. So for example, we can take some gene expression measurements from a sample and say, give me chromatin conformation. And the model says, oh, OK, well, the organization of chromatin is associated with some type of gene expression. And I'm going to spit out this chromosome contact map. And so what are some desired properties of this model? We'd like it to be able to input and output all types of high throughput data that we can imagine. So that means images, sequences, counts, affinity maps, so time series, traces, whatever. We want to be able to have sparse input, so we can take a subset of these things and query the model. And we care about this very, very poorly defined thing, but everyone cares about it, called interpretability. If we actually knew what this was, we could build an objective function for it and add it to the model. But we don't know what it is, so we're gonna find some heuristics of interpretability. So no matter what we put into the model, it will all be embedded into a common representation space, so that I can take this high C contact map, and I can take an image of a cell, and I can say, how far apart are these two cells? Even though I have two totally different types of measurements. We can introduce information theoretic approaches, so we can give the model a very, very large state space for these cells to live in, and it instead compresses the data into the smallest possible, the most compact representation. And we can also account for both uncertainty in the data, so the uncertainty between two types of relationships, and as well as uncertainty in the model. And if we built such a thing, which I'm trying to convince you that we can, or I will try to convince you that we can, what could we do with it? So we could, for example, determine relationships between arbitrary measurements. OK, well, cell shape is regulated, or at least strongly associated with these genes. Figure out what the minimal measurements we need to perform to be able to estimate the rest of a system. Build in a probabilistic interpretation, so we could say, oh, is this observation expected? Is this usual or unusual? And we can also use it for experiment design. So what is the experiment that I should run to best improve my model? Or what are the drugs that I should give to this cell to recover a phenotype that I'm interested in? OK, so I've told you what this thing is. And now I'm going to tell you how we're going to build it. So there's three parts of the Babel fish. There's the input, there's the digestion, and the output. Oh, and did I tell you we're going to do this with neural networks? That's great. Neural networks are amazing. So as far as the input goes, we have many types of data, and it takes different forms, right? We could have matrices, vectors, tensors, and all of these things have different types of properties. And the technology exists now, or even like point clouds, technology exists now to take any of this data and compress it into a space of fixed dimensionality. So we can take images of different sizes, compress them into the same size space with convolutional neural networks. We can take sequences of varying length, compress them into a fixed dimensionality space with recurrent neural networks. Because neural networks allow us to do anything that is differentiable, we can perform matrix factorization to find low-dimensional representations of elements for different types of structured matrices, and so on and so forth. And so we would call these, for a particular type of data, you would say, oh, I need to find a compression head. So one of these different types, and this is not a complete list, one of these different types of transformations to take my data and put it into a common space. Okay, so if I have an image, fish counts, an experiment, and like my description of an experimental condition, I find my compression head and compress all of these things together into a representation of fixed dimensionality. Then I need to perform some sort of operation that is sort of like given an arbitrary permutation of these inputs, I get the same result, so I can just add them together. Put this through another neural network, and then get a low-dimensional representation, hopefully, of everything that I know so far. And we can make this a probabilistic or a variational representation, so we output a representation that is the mean and standard deviation, or like level of uncertainty of that representation. How do we get an output? We take that representation, which is again the representation of the cell state, the universal representation of cell state with levels of uncertainty, and we can give it a query. So the query is, give me the 3C contact map. That gets introduced into the model, so we're sort of building a conditional model. And then find the decoder head to go from my dimensionality of my representation to output that type of representation that I want. So this would be, for example, a structured matrix that represents the contact map of the system, given the observations that I have. So hopefully this would come out. It would be great. So now I've told you how we built it, and now I'm hopefully going to convince you that the parts of this fish already exist in the universe. Okay, and all we need to do, at least what we're going to try to do for the next 5 years, is stitch them all together. There's been some very interesting research recently about modeling uncertainty. So here are some images of street scenes. This second column here is a, quote, ground truth segmentation of those street scenes. This third column is what the model believes is present in the scenery. And then there are two types of uncertainty that the model also outputs. A level of uncertainty given the data it's observed. So maybe these things, you could call these noisy labels, and the model is trying to recapitulate how noisy this observation could be. And then the other level of uncertainty is uncertainty in the parameters of the model. So the model is self-aware and says, oh, I don't know, this parameter could vary and I could get the same result. Let me tell you, where I could probably get more data and improve my prediction. There's image to sequence models where we can input an image of, for example, a cell, and it says, or instead of a cell, it's a dog, and it says, the model outputs dog plays catch with white ball near wooden fence. And this exists. This is 3 years old already, and the opposite thing, we can do the opposite thing as well. So you query the model and you say, I want to see an image of this bird is white with some black on its head and wings and has a long orange beak. And this is the output of the model. This is an image that doesn't exist in the real world, and the model has some representation of this sequence and a mapping of this sequence into an image space. And you can do this for very, very complicated things, like this flower has overlapping pink pointed petals surrounded a ring of short yellow filaments, and the model outputs this, which is a pretty approximate representation of that sentence. Google, for example, has built universal translators, and very interestingly, they built a model where they took English to Korean sentences and English to Japanese sentences, but not Korean to Japanese sentences. So there's incomplete A/B transformations. And what is shown here is 3 sentences with the same syntactic meaning, but in 3 different languages, and they're all embedded in the same representation of this model. So the stratosphere extends from about 10 kilometers to 50 kilometers in altitude, and those all form a cluster, even though those sentences are represented in 3 totally, or 2 similar and 1 totally different language as the connector, and that they all localize in the same space. Just so you guys don't think I'm totally nuts, we've built this sort of thing as well, where we can take an image, a reference image of a cell, find it's a low-dimensional representation, say, I want to see a particular structure inside of this cell. So this is a query system that we've built. And then we have this other variable, which is a representation of uncertainty that we can explore. And the model outputs a particular subcellular structure conditioned on this information and the query. And so this is I think, like alpha-actinin. And we can say, oh, I want mitochondria, or I want the nuclear membrane, and so on and so forth. We've actually done this twice with 2 different types of models. Here, this is an image of a bright field image of a cell. So this is just shining white light through the cell. And we say, show me, given this image, show me where DNA is inside of this image, or show me where the cell membrane is, or the nuclear envelope, or mitochondria. And this model was trained only on pairs of 1 bright field time point and 1 fluorescence image time point of the same sample. And we can apply these models to a time series of bright field and observe dynamics that we would never be able to see in live cell imaging. So what's happening here is that this cell, which I'm sure you guys can see in this bright field image, is undergoing mitosis. So the chromatin is condensing, and there's a division event. The cell membrane forms 2 cells and breaks apart. The nuclear envelope breaks down and then starts reforming, and the mitochondria become redistributed and when 2 cells are formed. And this is well studied, but this has never been shown all together in one sample at the same time. So of course, there are plenty of barriers to this. This is a non-trivial data munging problem. All of this data exists. We need to get it together in the same place so that we can use this to train a model. Deep learning or neural networks are data hungry. We need as much data as possible to get good results. The representation and interpretability of these models is always a hard problem. People always want to know why, how does it work? And there's plenty of research now, and more upcoming interesting research will certainly happen in the next 5 years of how to pull these models apart and see how their insides work. But other than that, everything that we described can be done right now. This can be done now. I have a variance of this model baking on some GPUs over in Seattle. And I hope I've convinced you. This future is just getting started. We're hoping that this type of tool will shorten the distance between biological fields, contextualize results in terms of adjacent fields, and be able to build us a testable hypothesis generator for how the components of biology or the languages of biology work together. So I'd like to thank you guys again for allowing me to present. And this is, as of the middle of summer, our team at the Allen Institute for Cell Science. I think we're like 75 people now, and we're growing. And if any of you guys are interested in working on this sort of thing, we are looking for PhDs and engineers to help us out. So thanks again.
Any questions? Yeah.
Hi, great talk. So you said you have some versions of this baking, because you showed an example of the translation for the fluorescent images.
Yeah.
Do you have versions of this? So this is a two-part question. Do you versions of this with a couple different tasks?
Yeah, so one of the models that we are testing out right now is taking arbitrary, so let me go back. So we have these simulated fluorescence images, or generated fluorescence images, and we now have like 20 channels. And so what we can do is we can run a simulation where we can take a subset of these channels and say, can you predict the rest of them? So that's a sort of a simulation of this idea. And we've also tried models from the Human Protein Atlas where we can take like amino acid sequences and say, and an image of a cell and say, where does that protein localize inside of the cell?
And secondly, my concern is when you're doing this for a lot of different tasks and you have all these different compression heads, is each one trained independently? So you talked about a common embedding space. Yeah. Presumably, so they're all the same dimensionality, but I think each of these models, these compression heads, are they trained independently to compress that specific data? Or is there some way to train this whole thing end to end so that you ensure that the embedding space, you can relate them between these different modalities?
So I think we're going to try the first approach.
End to end?
Well, with training the individual heads separately for certain types of modalities.
Okay.
But if you have a better idea, I'd like to hear it.
So I'm just curious as to how then, if they're independent, how then you would be able to relate their embedded spaces.
Oh yeah. So when we train the model, we'd be training with all of these data types simultaneously across all of the heads.
Okay.
And we can build sort of like autoencoders and things like that to make sure that, with different constraints, to make sure that there is a common representation space depending on what head you use, or independent of what head you use.
Fantastic. But you want this to poke holes, right?
Yeah, I'm absolutely hoping that
we'll have lunch together. I have a lot. Maybe to give you an opportunity here on that paper, I haven't read the paper yet. Congratulations. Of course, you guys pick again channels which are sort of, and don't misunderstand me, but which are trivially linked with each other, right?
Yeah.
Have you now done that experiment and imaged, let's say, DNA, nuclear envelopment mitochondria, and really tracked quantitatively in the same cell these features, and then shown how close is your predicted model?
So our assay development team is working on that right now. And so,
okay, so you got it published without that validation?
We have correlations, but we don't have, sort of people going in and counting things. And those relationships definitely vary depending on what we're trying to predict.
Right.
And I think that is an important thing, because that tells us, what is the coupling between these two types of measurements, which is what we're trying to find out.
Right, and then if I may add one thing, and that's maybe the first real big hole in the whole thing. I'm really not challenging the frame. I think someone like you guys should do it, right? I'm just not so hopeful that it will satisfy me as a quantitative biologist.
Yeah, well, I think one of the important parts is that maybe that prediction, the output of such a model, if we can build it, that prediction is not the ground truth. That prediction allows you to say, what is the experiment that I think is the most valuable one to run, given the knowledge that I have.
Right, but I wanted to add one sentence.
Okay.
It's fine, you interrupted me, no problem. If I do now that experiment, right, DNA, nuclear, or mitochondria, right, and I measure, I love measuring things, right, and I get a set of measurements which are going to be different than your measurements. The question now is how physiologically relevant are these differences, right?
Yeah.
And I think that's where it gets really, really hard.
Yes, totally. And
because you will produce wonderful artificial images, which look to my eye perfectly satisfactory, right? But the question is it meaningful for the physiology of that cell?
And I think this is a problem with any type of predictive model. So, yeah. Looking forward to lunch.
I think we're unfortunately out of time, but Greg will be around to talk more, so let's thank him again.